I love getting into a specific topic like that and then drill down and you just read all the literature and eventually you start to see the story. I'm always looking for the storyline. And it's a storyline that nobody's told before. So it's really fun that way. It's like a puzzle to solve, and I love that.
Hey everybody, this is Victor Cazzetto, and you just saw a clip of me and Dr. Stephanie Seneff speaking about hydrogen and deuterium, glyphosate, autism, a whole bunch of other things. What I'm showing you here right now in the background is some information on hydrogen, just to emphasize how important it is for your health because it makes up most of your body. Dr. Seneff has books published, papers published. She's a senior research scientist at MIT. She's amazing. You want to listen to what she has to say. Not only do we talk about hydrogen and heavy hydrogen or deuterium, but we'll touch upon autism, we'll touch upon Parkinson's disease, and a whole bunch of other things. Hydrogen is very important. Listen to more clips of my interview with Stephanie and then enjoy the entire interview. It is really worth listening to. And thank you, Dr. Seneff, for doing this second interview. And we will be doing more stuff together. Looking forward to it. Enjoy, everybody.
I suspect, and I don't exactly remember everything we talked about, but I suspect we didn't go into some of the details about the Parkinson's disease, because that's pretty new.
Yeah, we did not.
Because that's a paper that followed. That's our most recent paper. We're actually working on 2 more right now, so we're really on fire. But
the discussion of deuterium and your research is helping us to understand why the dirty stuff is so bad.
Yes, exactly. It's insane. I mean, can't they just see it's not working and just forget the whole program and do something different?
Yeah.
That's what we need. I wish that the government would take away all the subsidies from these big farms.
Yes.
And then give them to people to get them to small family farms, regenerative agriculture. Give the money to them,
100%.
I think just to give people a little bit more background, deuterium is heavy hydrogen. It has an extra neutron. It's twice as heavy. It's a natural element. It's all over the place. We've got lots of it in our body. And the problem with deuterium is that it can mess up the ATPase pumps. It's like putting sugar in the gas tank.
Yeah, it's interesting because if we, just by reintroducing higher nutrient levels, explicitly with the B vitamin, which would be a normal thing in a proper, clean, healthy diet, we would not even think about MTHR and
I know, yeah.
We wouldn't, it wouldn't be a consideration at all because everyone would have enough B vitamins.
Exactly. And I think even if you have a version that's not quite perfect, it would be fine, except that you've got the poison on top of it. It's really scary because it's so easy to hype a product and then to make money off of it, right? So that's the thing. And I don't think the naturopaths that are selling various supplements are a whole lot better than the pharmaceutical industry that's selling drugs, right? It's kind of the same game.
You're 100% right. Exactly. And this is my greatest fear.
I'm so pleased that finally the government is actually acknowledging that we have a problem. It's a huge step just to say we have a problem,
Right.
Yeah. So they're making it visible and making public awareness of the problem by virtue of the government acknowledging that there is a problem. Fixing it is going to be really hard. And I've been really watching the glyphosate situation because, Bayer is threatening to withdraw glyphosate from the market. You know about all that, right? I think we may have talked about that last time.
I would love that if they withdrew it from the market.
I know. I'm like, great, do it. But then the states are terrified if they can't get glyphosate. People are going to starve. I mean, it's just like we can't feed our nation without glyphosate. And meanwhile, there's all these weeds that are failing, that are getting smart, and so they have to use other chemicals besides glyphosate. Just this morning, I read an article boasting about this 3-way, triple stack.
I saw your comment on that, or did you mention that in one of your papers?
I don't think so, but I did put out something on Twitter today. I was kind of busy this morning twittering for Xing, I should say. But they had this article. It's a published peer-reviewed article that talks about, oh my God, this is so fantastic. We've got these triple stack genes, so GMO technology to give them resistance to 3 different toxic chemicals, which are all herbicides: glyphosate, glyphosate, and dicamba, all 3 for the sugar beets. And so that is just so insane. And then in that article, they said, well, already there's lots of resistance to dicamba. There's lots of resistance to glyphosate. It's worrisome that this might not last very long because they'll develop resistance to glufosinate. Then maybe they'll have to put some 2,4-D in there. I don't know, but they're going to have like quadruple, quintuple resistance. It's insane. I mean, can't they just see it's not working and just forget the whole program and do something different?
Yeah.
That's what we need. I wish that the government would take away all the subsidies from these big farms.
Yes.
And then give them to people to get them to small family farms, regenerative agriculture, give the money to them,
100%. I think you alluded actually what you said about, they're worried that the states are worried that their farming is going to fail and they're not going to be able to feed the people. That's the solution. Let those big farms fail because the small farms feed the world.
They'll take over and it'll be such healthier food, We really need those farms that are messy, that have all these different trees, some fruit trees and and then some zucchinis. I mean, just various kinds of crops that are all really healthy foods instead of just pouring, sugar and you get sugar and starch and protein, that's been soy protein bars. I mean, that is so synthetic. It's just disgusting food.
Yes, it makes me.
And get rid of it, just get rid of it.
100%, 100%, absolutely. I was going to say there was, the last interview I did with you, I took a clip from it and put it right in the beginning.
Yes, that's clever when you do that. I like that.
Right. And because you just said that, look, it just clean living. That's all we need to do.
Exactly.
It's so simple. And this is the beauty of Maha too. It's just clean everything up. But clean the air, the water, the food, the regenerative farming, preserve our habitats. And you're done.
Absolutely.
That's like the.
And you just have to really teach the people. I mean, forget about the regulation. I mean, Trump is not big on regulation. He wants to draw them back, right? So he's not going to say, oh, you can't use glyphosate or you have to be registered. It has to be a registered chemical. I don't think he's going to do that. All we can do is teach the public this stuff is poison. If you want your kids to have autism, fine, eat the food, It's like you just train them and then they can say, oh my God, I switched my family to organic and they're so much healthier. Tell their neighbor, tell their family, spread it by word of mouth and then stop buying the stupid soy protein bars.
Right. Absolutely. 100%. I started to do an autism working group on Wednesday nights.
Oh, nice.
With my focus being on the fact that we can help these children. They're just being poisoned.
Yes.
Because everyone's been brainwashed that, oh no, it's a genetic defect or this or that. Hello.
Oh, I know.
Yeah. I was like, look, genetic just factors into how we have to detox, but no, these are not defective children. They've been poisoned. Yeah. We can clean them up, period.
Right. It's funny because they've spent so much money on looking for the genetic cause of autism.
I know.
And they have found hundreds of genes and each one is like, less than 1% of the autistic cases. It's like such a mess. How can you possibly make any use out of that?
I think it's just about the money. It's just another.
I know. Well, genetics is a safe thing to study, right? You don't want to look at vaccines. You don't want to look at glyphosate, just look at genetics. It's very annoying to me that they spend all this money on genetics. I mean, it's obviously an environmental disease.
Yep. Yeah, absolutely. I mean, like I said, I love that clip because that's the answer.
Yeah.
Healthy living, eat clean.
And what can be wrong with that? Go out, get some exercise, get some sunlight, breathe some fresh air. I mean, it's just simple, right? Really simple.
And and now I'm going to try to spin us over to the deuterium.
My favorite topic.
A little bit because, so the cool thing is because people, like you said, we have to educate people. So the discussion of deuterium and your research is helping us to understand why the dirty stuff is so bad.
Yes, exactly.
It's giving us more insight. This is another reason why GMOs are terrible, etc. And this is why I've been excited about deuterium too. And of course you've also elucidated some other really cool things. This idea of sequestering, how it can be used in a positive way, etc. I really did. I loved the first interview. There was a lot of cool stuff that came out.
I'm so excited about deuterium. I'm so thankful to Laszlo Boros. I want to mention a shout out to him again because he reached out to me blind December 2019. I remember it so well, right before COVID hit.
Wow.
you know, and he was congratulating me on a paper that I'd written together with Greg Nye, which was a fun paper. We looked at and we actually talked about glyphosate in that paper, but it had to do with cobalamin issues with cobalamin absorption, and then pathways that were involved with sulfation. It was a complicated metabolic story that we worked out that kind of showed how the body readjusts, you know, so as to in fact mess up the brain ultimately, because of all these issues that come about because of glyphosate. So it was a good paper, but he saw that paper and commented on it. I said, gee, that's a great paper. And then he said, oh, by the way, deuterium. And so you know, I didn't know anything. I knew what deuterium was. That's about it. You know, I had no idea that it had any role in biology. And I just jumped on that right away. It's funny how I've had these moments in my life when an epiphany happens and it just completely changes my direction of research in a positive way. It's just been quite an interesting personal history, you know.
I think that's one of the great, and you've made some really huge twists. I mean, from computer science to biology, or from biology to computer science and back to biology, right?
Yeah, biology has always been my first love. And even when I was a computer scientist, I was reading, you know, just for pleasure, I would read books in biology. So I've always been kind of a biology nerd, or, you know,
right.
fanatic.
I mean, we got a bit of physics mixed in here when we start really getting deep into deuterium.
It does need physics, and I wish I knew more. I actually majored in biophysics, so I took several physics courses as an undergraduate. But I don't feel like I have enough physics because it's hard stuff, you know, these resonance frequencies and whatnot.
You're still young, you know, so
I know. I have to go back to school.
there are other things going on with deuterium and physics and like quantum deuterium crystals.
Exactly, quantum physics. I mean, my eyes glaze over. I can't really understand quantum physics. I guess I have to be honest and say, although I think it's really, really interesting. So
I think when I think it's just about passion, right? So if, you know, like if you come across something that suddenly catches your interest, you'll probably end up becoming an expert in quantum physics if you want to.
Yeah, I would like to. I admit that I still find papers on deuterium and I read them, but I realize I'm missing a lot because of a lack of background, and it kind of really frustrates me, you know.
It goes down a different rabbit hole. But
yeah, I keep thinking I might be able to just read enough and finally get it, but it's hard stuff, you know, and it often just I just don't get it really at the basic level.
This is like really the bleeding edge, you know, and like you've just had several papers this year.
I've done really well this year. Yeah, I'm really excited.
3 of you were right. There were
we think Anthony Kyriakopoulos, and Greg Nye and me. The 3 of us have had a lot of fun collaborating. Right. Yeah, Anthony's very, very brilliant. He writes stuff that's sometimes too hard for me to understand. So wow. I have to decode it and then rewrite it in a more accessible way. And then Greg will take a run at it and simplify it even more. And so we make a great team that way, because Anthony's really deep in the science and I'm in the middle. And then Greg he's a clinician and he's a smart guy and he knows a lot, but he knows how to make it more accessible to people who may not have the background, which is important because we don't want to just be right over people's heads, you know.
Well, we can't geek out too much. Well, yeah. I'm hoping to even bring it down another notch. So
yeah,
which I hope I could do. And so today too, like I know we may have mentioned taurine in the last video too, and I know
taurine's an interesting one. Yes, and we wrote a long paper on taurine, the 3 of us. Yes, that was fascinating. I love getting into a specific topic like that and then drill down. And you just read all the literature and eventually you start to see the story. I'm always looking for the storyline. And it's a storyline that nobody's told before, you know? Mm-hmm. So it's really fun that way. It's like a puzzle to solve. And I love that.
Yes. Yeah. And I enjoyed that too, because we had touched on a few things, you know, like I'm big on like the MSM and the gut biome and the kefir. And we had talked at the butyrate.
Absolutely, the butyrate. Oh my God, there's so much on butyrate these days. They're really recognizing that that's a key cornerstone of our lack of health is inadequate butyrate. I really believe that.
And deuterium is part of that picture.
It very much is. Yes.
So since we spoke last time, the other video we really did touch upon almost all of these things. But is there something in the last 4 months since we spoke that we didn't, that you can think of off the top of your head that we
I
didn't?
suspect, and I don't exactly remember everything we talked about, but I suspect we didn't go into some of the details about the Parkinson's disease, because that's pretty new.
Yeah, we did not.
Because that's a paper that followed. That's our most recent paper. We're actually working on 2 more right now, so we're really on fire. But we've got one that's getting close and one that's just started. So we're parallel processing our papers. It's just like amazing how much we have to write about. I'm very much like a kid in the candy store right now. It's really fun.
Oh.
Parkinson's is really, really interesting. And I became, yeah, I get a hunch sometimes, about something, and then I want to see if I can get the science to support the hunch. It's kind of how I work in many ways. And deuterium has been so interesting because, just to give people a little bit more background, deuterium is heavy hydrogen. It has an extra neutron. It's twice as heavy. It's a natural element. It's all over the place. We've got lots of it in our body. And the problem with deuterium is that it can mess up the ATPase pumps. It's like putting sugar in the gas tank. And then the pumps don't work well. They don't produce as much ATP. They become inefficient. They spew out reactive oxygen. So it's kind of like an engine not working is sort of releasing a lot of toxic gases, right? It's a similar concept. It's an engine that's not running well. And so it can't make as much energy, and it can't, and it releases this reactive oxygen that can then damage the mitochondria and even damage the whole cell. So that's a big problem. And so the organism knows that, at the basic level, it knows that. And there are all these processes that are in place in its extremely sophisticated biology that targets keeping deuterium out of the mitochondria. It's almost like an obsession with metabolism, I think. Keep deuterium out of the mitochondria. And there are many enzymes involved and many proteins involved and other things. I mean, it's just amazing how well that works normally when things are going well. A big issue is that I believe glyphosate suppresses many of those enzymes that do that job. And that ends up with mitochondrial dysfunction. And that's a key. There's several papers that talk about glyphosate causing mitochondrial dysfunction, although they don't exactly understand how. I think the how is simply that there's too much deuterium.
Right, more than the body can handle, right? So the body is designed to handle a certain amount of deuterium. But
and even it's good for you. I mean, because if you can put it in the bones, it'll strengthen them, for example. So the body tries to sequester the deuterium in places where it can even be useful. It's kind of like iron. Iron can be both toxic and deficient.
Right.
So it's the same idea.
Right. Right. There's that expression. I mean, the definition of a toxin is just quantity.
Yeah, it's really interesting because, and actually all the metals are like that. They can be toxic. Well, some of them are only toxic, like aluminum, there's no good level of aluminum and there's no use for it in biology. Lead is like that too. It's totally bad. And of course, mercury, there's no use for mercury. So aluminum, mercury, lead, these are and probably cesium, these are really toxic, and there's no good use. Metals. But there are all these micronutrient metals like minerals, magnesium, manganese, zinc, iron. They're all essential, because certain enzymes depend upon them to work. Selenium is another one. But if there's too much in the wrong place, it's toxic. Because the metals are so reactive that they can cause reactions that you don't want if they're not kept sort of packaged up and safe. And there's these chelators that can actually transport these metals. And there's a whole system in place that delivers them. So you have to catch it, take it somewhere, deliver it safely, and then it arrives at the enzyme can use it. There's a whole process involved that's complicated with lots of support from various enzymes and stuff. So nothing is simple in biology, that's for sure. But when you start messing up some of those proteins, you get in trouble you can get simultaneous toxicity and deficiency, which is what I think you have with deuterium because of glyphosate.
It's triggering a whole bunch of other problems. So you're just speaking about how like the deuterium can trigger a bunch of other issues, right? If we cross the threshold of what we can handle.
Yes. So the Parkinson story is really super interesting. And it started with, I was looking for ways to sequester deuterium because I realized there were certain interesting molecules that potentially could grab hold of deuterium and hang on to it for dear life and never let it go. And that becomes a mechanism for keeping it away from where you don't want it to be. And so that could be a way to kind of shrink the levels in the blood by virtue of trapping deuterium atoms one by one inside these sort of cavities that won't let go. And so I've had a lot of fun with that. And we actually published a paper on some carbon-based molecules that have specific carbon atoms in specific interesting places that have this property. And you learn it from the work they do with, they like to put deuterium into molecules because they can trace it. And this gets into this quantum physics stuff where there's these vibrations and they can detect the deuterium. And there's some interesting opportunities to figure out how proteins are shaping and stuff like that. They use that for research. And what they'll do is put the protein, for example, the peptide sequence, into heavy water. So there's tons of deuterium. They put it into heavy water, and then some of the atoms, you have the carbon, the sulfur, the four key ones are nitrogen, oxygen, carbon, and sulfur. Those are the big guys. And they have different degrees of willingness to let go of their hydrogen and replace it with the deuterium. And so the oxygens and nitrogens, they grab it a hold right away. If you have it in heavy water, they just, okay, get rid of my hydrogen. I'll just swap it out for deuterium. They do it right away. The carbon atoms are more stubborn. They mostly don't. And in fact, if you put a peptide sequence into heavy water, most of the carbons just hang on to their hydrogen, don't let go. But there's just a few carbons that will swap out their hydrogen for deuterium. They don't do it as fast, but they do it. And then if you take that molecule now, put it back into regular water, all those oxygen and nitrogen atoms, they let go immediately, get back their hydrogen. Like they let it go. But that carbon atom that grabbed it won't let go. It'll hang on to it and won't even in very highly acidic conditions where you would expect to be a tremendous opportunity to swap out that deuterium and put back protein. They don't do it. And so this is the histidine. Histidine is an amino acid, and the imidazole ring has that property. It's so cool.
That double bond, the cis bond.
Yeah, there's the histidine has this ring that has a carbon that has its neighbors are nitrogen, both nitrogen, and it's in a ring. So that's a very unusual situation for a carbon atom. It's unique to histidine among all the proteins. Mm-hmm. And histidine is the only amino acid that picked up deuterium when you put it into the heavy water, the only one. So this starts to make me think, okay, if that can happen, maybe biology knows that, and they're going to want to find a way to put a deuterium there and hold on to it forever, basically. As long as you've got that imidazole ring, you've got that deuterium trapped. And biology may use that as a way to trap deuterium. So that was one thing I was thinking. And then there's also these bisallylic carbon atoms, which we probably did talk about before.
Yes.
Those are fun. Yeah. And so this paper talks about both of them, the histidines and the bisallylic carbons. The bisallylic carbons are in polyunsaturated fatty acids. So DHA, for example, which is like you take fish oil, it's a popular EPA and DHA are these long chain, highly unsaturated fatty acids that are considered to be healthy, although they're very reactive. Those are the ones that will cause these chain reactions that will damage everything if they get going with the reactive oxygen. So you don't want to have a lot of reactive oxygen lying around if you've got a bunch of these lipids. But on the other hand, if you do have a lot of reactive oxygen, maybe you can trap deuterium. And so I suspect that might be going on in the membranes which are enriched in these bisallylic carbons. Those guys are the ones that react and they cause all these different products, eicosanoids, and there's all kinds of different names for these products of the fatty acid oxidation that are signaling molecules. Actually, they have powerful signaling capabilities and whatnot. And there are even products called resolvins that actually tame the reaction. So some of the products make the reaction bigger, inflammatory reaction. This is an inflammatory response. It's kind of like having a fire and then eventually putting out the fire. And the resolvins will put out the fire, and they actually quench the chain reaction. Well, the deuterium can quench the chain reaction if you can put it there. And that is just so cool. So I think perhaps that all of this stuff that goes on with membranes, inflamed membranes, which of course is like, joint pain, back pain, all these things that have arthritis, all these things have like inflamed joints. There's membranes that are being attacked by oxygen, reactive oxygen. And then all these different products are being made by these various enzymes even that can do this. And then signaling is going on. And then eventually you start getting resolvins showing up. These are reaction products that are a redirect towards a different enzyme because the other one stops working once there's deuterium. is so fascinating. So they've shown that in papers that if you put deuterium on these reactive carbon atoms, which are these bisallylic carbons, if you load them up with deuterium, they don't react. That enzyme doesn't work. It has what's called a high deuterium kinetic isotope effect. Mm. And so it goes down a different pathway, which produces these good guys, you know. It's really fascinating. So now I'm sort of thinking that when you have a membrane that's on fire, you're making all these products and you're doing all this signaling. And in the meantime, you're capturing deuterium atoms. So you're cleaning up the deuterium out of the medium by capturing those atoms. And then you permanently capture them on those lipids. And now you've got a problem because you need to sequester those lipids. You want to sort of tuck them away and let them never go anywhere, right?
Where should they ultimately end up?
Well, they may ultimately end up in amyloid beta plaque and in Lewy bodies, you know, all these different debris that you see in the brains of people who have disease. I think it's possible that this was what I was thinking originally, was that maybe the plaque has a purpose in trapping deuterium and getting it out of the way so that it won't mess up the neuron. And that's basically the theme of this paper that we wrote on Parkinson's disease, which is available as a preprint. But we have a version that is under review and it's looking good. So I'm hoping it's going to get peer reviewed. We'll see. But the idea is that the membranes, and this gets into the mitochondria themselves, because actually it looks to me like there's all these different amyloidogenic proteins that can misfold and cause trouble. Mm-hmm. And they form these fibrils and then they form these deposits. But it's looking like, and you know, amyloid beta, for example, in Alzheimer's disease. I'm getting a little bit all over the place here, but it all does kind of tie together.
Related. It's all related.
Yeah. So if you look at amyloid, and I've been reading about these neurodegenerative diseases for a long time. I'm very interested in them. I'm fascinated by them, actually. And with Alzheimer's, you know, they've been spending all their money on Alzheimer's treatment, trying to come up with antibodies that can trap the amyloid beta and keep it from forming the plaque. You know, they were completely hung up on, the plaque, it's obviously there. That's Alzheimer's. We got to get rid of the plaque. How can we do that? We put through these antibodies. And they've done all this research and they've got all these things and they find things that actually do, you know, work as antibodies, but they don't help the Alzheimer's. And in some cases, they make it worse. So they're like, finally, they're saying, well, maybe the plaque's not the problem, you know, right. Maybe it's something else. And then, so there's a lot now. You look at all these misfolding proteins and you look at the most recent papers on them, and you will see that they're saying, well, maybe it's these oligomers, you know, these soluble oligomers. Maybe those are the problem, not the plaque, you know. And I think that's right, the oligomers. And then it turns out that the lipids in the membrane, certain lipids, and in particular lipids that have a lot of these bisallylic carbon atoms, they actually can cause these proteins to misfold in a way, forming these oligomers that then bore into the membrane and dig a hole. It is so interesting. And it's true for, I've learned about, found papers on multiple of these misfolding proteins that talk about that possibility, that they can form this oligomer that then goes into the membrane and makes a hole in it, forms a pore. And then that can even allow them to get inside the mitochondrion. Oh. And then the mitochondrion has this extremely unusual lipid called cardiolipin, which mostly hangs out in the inner membrane where the ATPase pumps are. Mm-hmm. And it actually completely surrounds the ATPase pumps. The cardiolipin is all over the place, right there with the pumps. And there are papers that say maybe the purpose of that cardiolipin, a purpose, is to trap protons as they come through to buffer them. There's actually papers that talk about buffering the protons. So my thought is, well, maybe they actually trap the deuterons as they come through. So you've got all these pumps and you don't want deuterium to get into them. This is your last chance to get rid of the deuterium. If you've got a filter in there that can actually grab hold of that deuterium and keep it from getting to the pump, that's fantastic, right?
Pump.
That would be the right place to put something like that. So cardiolipin actually gets put onto the inner membrane. It's made inside the matrix of the mitochondria. Mm-hmm. It's put into the inner membrane right there with the pumps. Maybe it grabs the deuteriums and sticks it onto their bisallylic carbon atoms, you know. So eventually, as the cardiolipin is sort of dodging bullets and grabbing deuterium, it eventually populates itself with deuterium atoms. And interestingly, if there's enough reactive oxygen in the mitochondria, the cardiolipin actually goes to the outer membrane. It moves, it migrates to the outer membrane.
Yeah.
And at the outer membrane, it can find those misfolding proteins in the cytoplasm. So once it gets to the outside world, there's the protein waiting to misfold. And it sees, oh my God, a cardiolipin, I better misfold. I better go in and drill a hole because this mitochondria is sick. I got to kill it, So it's possible that the whole thing is that the mitochondria are constantly being recycled. That's part of natural living. They get born, they do their job, they get sick, they get removed, new ones come. It's a constant recycling. So you need to figure out which ones are sick and take them down. And that's what this can do.
I want to give you an analogy and tell me if this is correct at the higher level when talking about the idea of like the plaque buildup and stuff. I think a lot of people are very familiar with candida.
Mm-hmm.
So the idea, like you were talking about, like removing the plaque, like a lot of people, they're like, okay, I got to kill the candida, kill it. And like when I work with clients, I'm like, no, you got to get rid of the heavy metals. The candida is there because of the heavy metals.
It's helping you to get rid of it, right?
Yes, exactly.
Yes.
So if you get rid of the heavy metals and then the candida goes away, right?
Yeah.
That's if you try, like you were saying, okay, like we try to remove the plaque, but it doesn't solve the problem.
Yeah, the candida might be getting rid of the deuterium actually, because yeast have a very an ATPase pump, that works in the reverse. It's not the one that's inside the mitochondria, but one that goes on the membrane. Do you know about this? On the yeast cell membrane, it's an ATPase enzyme that actually has to burn ATP. It uses up ATP, and it pumps protons out against a gradient. So it's the opposite of the ones inside the mitochondria. Instead of using protons as a motive force to generate ATP, right, it's actually using ATP as a force to get the protons out of the cell. And yeast cells can actually survive in a very high deuterium environment. It's really interesting. So when they push out, and it turns out those ATPase pumps that they have that are a different kind of ATPase, those are very reluctant. They have a very, very high deuterium kinetic isotope effect. So when they pump out protons, they don't pump out deuterons. Very few deuterons go out. So they concentrate deuterium inside that cell. So my guess is that candida probably is able to trap deuterium and keep it away from the host.
Another reason why we would have candida overgrowth perhaps is too much deuterium intake.
Yeah, and inability to manage deuterium properly is really what it amounts to, because you have to have very high production of hydrogen gas by the gut microbes, and then the recycling, using that hydrogen gas as a reducing agent with carbon dioxide.
It's depleted deuterium, that hydrogen gas.
Yeah, it's super depleted. And then you make these organic molecules and those are like the short chain fatty acids, acetate, propionate, and butyrate. Those three are super, super important in the gut. And that's where there's all these papers coming out now talking about deficiencies. In fact, glyphosate, there's a paper on glyphosate that shows it was a good study on mice, and they actually exposed them to different mice to different levels of glyphosate. And then they looked at how much glyphosate was in the gut and in the feces. They had these pictures that were extremely high, very low p-value, like p-value was 0.0001 for both the amount of acetate in the gut and the pH of the gut. The pH goes up when there's more glyphosate. And when the pH gets too high, the microbes that make the acetate don't do well. They don't thrive. So you end up with a deficiency in those microbes. And then you have a deficiency in butyrate, which comes from acetate. So the whole thing breaks down. You don't have enough of those short chain fatty acids, and you don't have enough of deuterium depleted nutrients, because that's what those are.
Right. Well,
it's really cool.
Kefir. Kefir is my cure-all for everything.
Kefir, yeah, that's great.
Butyrate production really was such a positive impact.
Yeah. And you can eat a lot of fiber too. Eating a lot of fiber is good because that really fuels the whole system, Right. But you need to have healthy lactobacillus and bifidobacteria. So
right, exactly.
Yeah.
It's amazing. And, hopefully more and more people are going to get back to this simple, healthy eating with some of these key foods.
Yes.
While we slowly clean up the world, reduce.
Right.
I mean, we're kind of in a race, right? Because
we really are. Yeah. Because in fact, I don't know if, when you have glyphosate in the mix, then certain enzymes aren't working, and then certain things don't happen, and then you can actually get toxicity. So if you look at sulfur, like we have sulfur deficiency, but if you eat, a lot of people have sulfur sensitivity. If they eat foods containing, say, sulfites, they get sick. And sulfite is very, very reactive. But the enzymes, there's plenty of enzymes in the gut that normally would just disappear the sulfite right away. Either take it down to methionine, for example, or take it up to sulfate, both of which are super useful. So the sulfite by itself is not good as long as it can be cleared very quickly by these enzymes that get broken by glyphosate. So now, if you eat the sulfur, you've got the glyphosate, you're going to harm yourself if you have sulfite reactivity, because the glyphosate won't let you clear. Same thing with formaldehyde, by the way. Formaldehyde is quite interesting, and I think glyphosate is really causing problems with too much formaldehyde in the gut and an inability to clear it. And actually clearing formaldehyde is part of making methyl groups, because formaldehyde is a precursor to methylene tetrahydrofolate, which gives you that methylene that becomes methyl with another. There's an enzyme, MTHFR, which is an enzyme that's quite popular among the autistic community because they all know about it. A lot of the kids have defects in MTHFR. That's the deficiency. Yeah. What'd you say?
It's a vitamin B deficiency. I always tell people when you have
I know, because it needs the vitamins. That's the thing. The B vitamins are getting messed up. It's amazing we're as healthy as we are, but you make the methyl tetrahydrofolate from the methylene tetrahydrofolate from the formaldehyde. And if that system's not working, the formaldehyde builds up, it's very toxic, But if it is working, you're making methyl groups, which are incredibly important. They get passed around in methylated proteins, methylated DNA from the methionine. And the methionine gets it from the methyl tetrahydrofolate. So it's a bit complicated chemistry, but you need those methyls for the methylation pathways. But glyphosate is messing up that system. So that's another way in which glyphosate is causing trouble.
Right. I think people need to realize too that it's, what you're explaining, this is like a 1-2 punch. It's a double whammy. Because you're not only missing the nutrient, but you've got this toxic buildup at the same time.
Yes, exactly. You've got nutrient deficiency, and especially micronutrient deficiency, inadequate B vitamins for sure.
Right.
That's something that Zen Honeycutt found when she tested the food. She's done a great job of testing different kinds of foods and school lunches at the public schools, and also fast food. She did 2 big tests, and she looked at, I think it was the school lunches. She found abysmally low levels of the B vitamins, all of them, abysmally low. And so that's very disturbing, because those are so important for mitochondrial health. They're used tremendously by the mitochondria.
Yeah, it's interesting because if we, just by reintroducing higher nutrient levels, explicitly with the B vitamin, which would be a normal thing in a proper, clean, healthy diet, we would not even think about MTHR and
I know, yeah.
We wouldn't, it wouldn't be a consideration at all, because everyone would have enough B vitamins.
Exactly. And I think even if you have a version that's not quite perfect, it would be fine, except that you've got the poison on top of it, and that cuts it over the edge. Right. So people have just a little bit of disability in that enzyme. It would have been perfectly fine, except that they had these exposures that caused it to not work. It was more sensitive to the exposures.
That's why, it's so interesting. The solution is really so incredibly simple, but we need to keep digging deeper into the science, to keep emphasizing to people why it's so important to eat clean and to clean the food, and eat healthy, nutritious food, not food that is devoid of nutrients.
Exactly. And it really is just organic whole foods. It's really quite simple. And as you say, fermented foods that contain sulfur, lots of fresh vegetables, lots of herbs. I think herbs and spices, I've really gotten to emphasize herbs and spices lately.
That's a magical trick that I also like to talk about a lot. So I'll do, you mentioned selenium earlier too. I throw mustard seed on food.
Yeah, great.
And I tell people, I was like, look, it doesn't have to be for the flavor. It's for the nutrition. That's like an easy way to get selenium from the mustard seed. So that people don't realize that the herbs and spices they're more than just flavors,
Yes, right. Absolutely. Yes. I've really gotten it, especially fresh herbs. When I was a child, I don't think I even knew that fresh herbs was something you could buy. We always had herbs came inside a jar, You'd sprinkle some oregano from the spice rack. I didn't know that you could buy them fresh. Here it's wonderful. We have these farms. They're so beautiful. We get a lot of these dill and oregano and cilantro. You know, we use those a lot in our cooking. They're so flavorful.
Yeah, yeah. It really makes a big difference. And then a lot of people can grow so many of the herbs in pots, you know, in their yards,
Yes.
their windowsills, even, etc.
Right.
It makes a big difference. So again, it's all about education. We just got to get the information out there. And so I really appreciate, I think the work that you do helps to motivate people. It helps people to understand why it matters to eat clean, eat nutritious, you know, because there are all crazy hidden dangers in the toxic foods. I mean, the craziness with the GMOs and stuff, those foods are sequestering more deuterium out of the environment and delivering it to our body.
Exactly. Yes. In fact, it's, you know, grass-fed dairy is lower in deuterium than the cattle cow dairy.
Right.
Yeah.
It's amazing. And, you know, little by little, we'll make deuterium a household name, a household word.
I really want to. I'm so excited about it, as you know. I'm trying to get the public aware. And of course, I'd love to get the government aware too, because Laszlo Boros is hoping that the government would, the present government might think about funding research, you know, to get more, because the research is grossly inadequate on deuterium. It's really frustrating. It's hard to even find how much deuterium is in different foods. There's a recent paper that was published that was good because they had a whole table and they had a lot of different foods, maybe about 20, I think, items. And that was more than I'd ever seen. I had just seen a handful and I wasn't even sure that it was peer-reviewed literature. It was just like an article on the web, you know. But this was a peer-reviewed paper that came out recently, and I'm sorry that I don't know the name, the title, or the authors, but I remember it had this table. And the table was fun because it had the ones with the higher deuterium were the sugars and the starches. And then in the middle, it starts to be the proteins, you know, and then at the bottom is all the fats. You know, the fats, on average, it's kind of starches, proteins, fats. So if you want low deuterium, eat fats, and especially animal-based fats.
Right.
And organic or grass-fed beef.
Yes. It really is worth it for us to make the effort to get the better, healthier, cleaner food. It makes such a big difference. Yeah, there's so much. So like I'm, you know, and I've mentioned before, like my big thing is about kind of going after the supplements. And
I know.
And it's so ironic, you know, we tell people eat whole foods, stay away from processed foods, but then people are running out to supplement with highly processed supplements.
Extremely processed individual amino acids, you know, in a pill.
Right. It's absolutely insane.
And
and I'm really concerned about that. In fact, I got to give another shout out to Zen because I've been trying to get her. I'm trying to speak to her about some of these products.
Yes,
some of them to her attention, because there's a lot of stuff out there too. There's a lot of products that are infiltrating the Maha movement that are they are not Maha.
It's really scary because it's so easy to hype a product and then to make money off of it, right? So that's the thing, and I don't think the naturopaths that are selling various supplements are a whole lot better than the pharmaceutical industry that's selling drugs, right? It's kind of the same game.
You're 100% right. Exactly. And this is my greatest fear. Like we've seen, this is what happens in industry. You shut them down. Those people just take their business plans, or other people copy their business plans, their business model, and they bring it to another industry.
Yeah.
The tobacco industry took over the food industry.
I know. Well, it's funny that people think, you know, oh yeah, processed foods, that's bad. But then they're popping these pills, which are incredibly processed. They're more processed than the processed foods, you know?
Exactly.
And that's fine. They don't seem to see the contradiction there. So
right. Well, I'm doing my best to try to educate people on that side. I know, who knows, maybe we'll do a video about something like that in the future if you're interested. But I know you've got a lot of things going on and your passion is
right
now.
We're having such a ball. I can't believe I'm sort of at the peak of my career right now. Most people have long since been retired, but it's a lot of fun. I hope I can keep my brain going. You know, there's so many people that are suffering as they get older from various kinds of dementia and whatnot. It's just
well, you sound pretty good to me, and you're living a clean life.
I know,
and I think that helps. You know. I think I wanted to ask you a question. We still have a little bit of time. So one thing, and I mentioned this before too, I love MSM and again, I don't like to recommend supplements, but MSM is something that's just in the rain. People like you might have an advantage. I don't know if you get out in the rain a lot.
I do, absolutely. Here in Hawaii, I mean, Hawaii is famous for these rainstorms that open up for 5 minutes and then the rainbow comes, you know.
So I think that's a huge advantage where most people, unfortunately, they've been so disconnected from that, and they're not eating the food that has been covered in the natural MSM from the rain, because we clean everything.
That's interesting. That's a really good point. I hadn't thought of that, actually. You just absorb it through your skin, right?
Yes.
It's like an Epsom salt bath.
Yes. So
yeah, that's really interesting.
And I see so many incredible recoveries and things just with very cheap supplements.
And DMSO is sort of a close cousin. That's
exactly. One. Yeah, molecularly, there's such a small difference.
They're very similar. I think it just has an extra oxygen.
Right. So there's one less on the
dimethyl sulfoxide and methyl sulfonomethane. So it's really just the oxygen that's different.
Right.
Yeah, they're both really interesting. And DMSO is used a lot in animals, I think, in veterinarians.
And used even more, I guess, kind of as a carrier, but it's really about like with the MSM, it's providing the methyl groups and the sulfur. And
I know, it's incredible, isn't it? Because both the methylation pathway and the sulfation pathway are in that molecule, which is amazing.
Yeah, it is. I've been in love with that for a long time. And it's related to a lot of the stuff you talk about, a little bit indirectly, but not so indirect,
Yeah, I know. I would love to see to trace the synthesis of biosynthesis of methyl sulfonomethane and MSM and DMSO, the biosynthesis. I actually discovered some microbes that grow in shallow seas. I may have talked to you about this recently. I discovered this article about these microbes that grow in shallow seas, and they make a product that's a gas. It's not either one of those, but it's like a precursor to them.
Oh, interesting.
It's a hydrogen. I'm not quite getting what it is. Dimethyl sulfide, maybe. Dimethyl sulfide. No, methyl sulfonomethane. Maybe it is.
Oh,
I think dimethyl sulfide would be almost methyl sulfonomethane, right? I think so. I think it's dimethyl sulfide. I believe so. That's really interesting because that's the one, you just get more oxygen with each step, right? Methyl sulfonomethane, and then dimethyl sulfoxide, and then dimethyl sulfide. It might be dimethyl sulfide.
Oh,
I'm going to have to go back and look because I don't think I registered it when I read it. But it's a gas, and it's produced by these shallow sea bacteria, specific species.
Oh.
And that gas actually seeds clouds. It causes clouds to form, and that and then over the oceans, huh?
I wonder if that's why the MSM is in the rain. There must be.
It could very well be. It gets oxidized. So it's the dimethyl sulfide that gets oxidized in the cloud or in the air.
That makes sense.
I think that totally makes sense. And it seeds the clouds, and then the clouds produce the rain, and then of course it's cloud cover, so you have less heat, and that keeps the oceans cooler. And that microbe is extremely sensitive to glyphosate. I found an article that showed it just gets completely clobbered by glyphosate. So you've got glyphosate coming out of the Mississippi River, killing off all these wonderful microbes in the shallow seas of the Caribbean and the Gulf of America, I should say.
Right. Wow. And then we end up with less MSM in the rain, which is detrimental for all of.
I know sulfur is. Of course, they had rules to try to get rid of sulfur in the air, and they did that a little too well, I think.
That's crazy. Yeah.
Yeah.
We need sulfur. Like you mentioned, some people, they can't handle sulfur. They say, "Oh, I can't take sulfur." I was like, no, it's that you can't handle certain sulfur compounds. We would die without sulfur.
Yeah, I know, exactly. And the thing is, if they avoid all the sulfur-containing foods, they're going to have sulfur deficiency.
Yes.
And that's really bad, because the sulfate is so important. I write a lot about sulfate in my writings.
I see that in a lot of your papers. There's always some sulfur compound.
Yeah, I really love sulfur, and I work on it. I actually soak in Epsom salt baths pretty much every day.
Fantastic.
Yeah.
I tell my clients, people like to do that. I was like, add a spoonful of borax.
Yeah.
Bath with a cup of baking soda. You'll pick up some boron.
Yeah, that's a good point. Yeah, boron is good too. Yeah, it's fascinating stuff, isn't it? The biology is just wonderful. It's such a huge package that you're never going to finish, so you're never bored. You've always got new things to learn every day,
That's a good.
which I love to learn. So that's really perfect.
I think that's the other thing too, and that I like to remind people is that no matter how technical we're getting, we don't know anything yet.
Yeah, this you can be blindsided. I still can have somebody introduce some new thing that I had no clue about that has a major role in biology. I'm like, how did I not know this?
Right, right.
Yeah, it happens all the time.
It's wonderful. That's why we have to stick around a lot longer. There's so much more.
I know it's a great hobby. You just never run out of things to do. So
And you think about like how powerful we are right now with all the science that we have. Think about how much more power lies ahead.
right, and of course the AI is going to help too. I think AI is pretty useful. You have to be careful, but you can really get if you want to learn about biology. It's so easy to just ask a question for the AI robot, and it'll explain something to you, and you can just self-educate yourself.
But it's also happy to miseducate you.
Oh, I know. I've seen some very big mistakes, so you have to watch out.
Yeah, we really have to be careful. I think, people still need to rely on others like yourself, to make sure that we know what we're talking about. So again, I appreciate the work you're doing, and I'm looking forward to seeing you continue to come out with more.
I'm
papers. So the Parkinson stuff, that's the next.
There's a preprint available on the web right now, and then I'm hoping the journal will accept it soon. I'm hoping, because we've already sent in the response to the reviewers, and it felt like we did a good job. So I'd be very surprised if they said, "Oh, sorry," I'd be surprised, but you never know. Maybe somebody else will come along. I actually had a paper like that where it was going fine, and we answered the reviewers, and then we got another reviewer pulled out of a hat, and the other reviewer hated the paper and wouldn't accept it under any conditions, and it got rejected. It was really amazing, and that had to do with cholesterol and statin drugs, that kind of thing, So they pulled somebody in from the industry to say, you got to get rid of this paper before it gets published. I'm sure of that, So I've had these kinds of experiences. I'm used to it.
Maybe we should do a video on statin drugs.
We could. I haven't done much about statins lately, but I do hate them.
Yeah, me too. Oh my gosh. I would love to do that because I'm always trying to discourage people from taking those. I'm like, no, please, you do not need these statin drugs, not at all.
Yeah, absolutely. So, I don't think anybody should take them.
Yeah, I agree 100%. Glad to hear you say that. You heard it here from Dr. Seneff. I got to give a shout out to one of my uncles, actually, who's incredibly healthy.
It's amazing. The doctors, they really put the fear of God in you, right?
Right. Yeah. Exactly. Yeah, I have an uncle, my Uncle Archie, who's great, actually, watches a lot of my videos, so he's probably going to be watching you, Dr. Seneff. And he's great. He's 88 years old. He looks fantastic, really healthy, eats tons of eggs. He's trying to do it. But the doctor scared him into the statin. I'm like, no.
I know, it's so hard if you're going to have any faith in your doctor. The thing is, you need to lose the faith in your doctor. I think the doctor does not know best.
Right. Until we educate them more. Right, we're going to be waking up a lot of.
Yeah, they're hard to educate because they've been so brainwashed by the industry.
Well, we're going to rely on the government to help slowly fix things, get out more appropriate information, expose the truth.
Yes, well, certainly RFK Jr. can do that. I hope he doesn't get too hobbled by all the opposition, but we'll see how that goes. I'm still very hopeful, but I understand that he has a very difficult road ahead.
Right. Yeah, it's a process. It's a long process. So the same thing. I'm remaining very optimistic. No matter how much opposition I see. The way I look at it, every time I see opposition, that's just a target for me. That's the way I see it. It's like, oh, okay. That's something out. They expose themselves. They need to be educated, guided, confronted, whatever.
Yeah.
So that's the way I look at it, because, they're in place, and wow, they are making great announcements.
Right. I know. I think they've done an amazing amount already in such a short time.
Oh, it's
and they've put a lot of good people in place too. I really like the NIH, Jay Bhattacharya from Stanford.
Yes.
Yeah. And I forgot. I forgot the name, but
I can't even keep track.
yeah, of course, Casey Means is interesting too as Surgeon General. Casey Means, she's not approved
I'm
yet,
a
but
little,
she's.
I'm a little bit concerned with that one.
I know. There's a lot of controversy around her, but I still think she's all for good, healthy food, and that's
That's
to me maybe the most important thing.
I think the other thing too is, when you talk about, you're going to have a group of people working together, and if we get one or two that we're a little concerned about, but they're surrounded by the group, so
Mhm.
should get
And it's not like she loves toxic food, I mean, she is exactly on the right place with respect to the
Right.
food. So that's already pretty huge to me.
No, but
You'd
it's
have to be pretty
connected.
bad to compensate for that,
Right.
to,
And that's the
yeah.
key. I mean, that is the key point. Like you were saying, we just clean everything up and eat clean, and everything else fixes itself.
I think so. It's nice that the formula is so simple, and everything that you could do to fix your health is not going to cause side effects. These are all good things to be doing in general anyway, just to, good living is what it's about.
Yes, 100%. And we'll all be healthier. All right.
Yeah.
I think I've kept you here for about an hour.
You did,
I'd like
yes.
to keep you here for another hour off air even, but
I know. It's such fun talking to you. It's been great. Thanks so
it's
much.
a pleasure for me. Thank you so much, Stephanie. Keep up the great work. Dr. Stephanie Seneff, Senior Research Scientist at MIT, doing amazing work on deuterium and many other things. People should go out and read your paper on
Parkinson's.
That's interesting. I got to go take a look at that. And we'll talk more in the future, I hope.
I certainly hope so. It's been great. Thank you.
I appreciate it.